Hey, so I think we should start today's live session. Uh, can anybody tell me in the chat whether I'm audible or not, or you can mute yourself? Hello. So hopefully I'm audible. Okay, it's audible. Okay, okay. Thanks. Okay. So I guess yeah, since it's already uh, time has passed quite a bit, 18 minutes, so it should start the session. Uh, okay, so again, I welcome all of you to the live tutorial session for week 4 for the course Bioinformatics Algorithms and Applications. The course, as you know, is instructed by Professor Michael Bromiha from IIT Madras. And I will take today's tutorial session. I am Sunanda Samantoy, PhD scholar at IIT Indore in the Department of Biosciences and Biomedical Engineering. So, in this week's content, we will cover some of the concepts related to what you saw in basically the main course content. For example, scoring matrices, position specific matrices, right? And also phylogeny, some concepts regarding that. So hydrophobicity, hydrophilicity, some concepts regarding those, and so on. So can anybody remind me, just as a brief recap, what we discussed last time, but in uh, last week's course and live tutorial sessions. So we used or we tried to understand about some of the alignment algorithms, right? So can anybody name what are those alignment algorithms? For global alignment, which algorithm is used?
Smith-Waterman algorithm.
So Smith-Waterman is for local alignment. So local alignment is where you basically need to find, let's say, sequence motifs, a set of sequence with particular motif, right? So those kind of things, and so basically subsequence alignment that is maximized by local alignment algorithms. For global alignment, we used Needleman-Wunsch. So if we remember, just as a quick recap, so what we had to do in Needleman-Wunsch is that, so what we had to do is that we had to calculate the scores for each of these grids, right? So if you notice on the top left corner, so there are some grids, and we had to put some scores on there. So how did you initialize the scoring? So okay, so let's talk about it. So let me get it done. Yeah, so first of all, we initialized with 0 at the top left corner. And then for each horizontal and vertical jumps, we just added 2 for the first row and the first column, right? So those will mean accumulate minus 2 in this case. Those will mean accumulating gap penalties, right? So as you see on the right hand side, it is written here that the gap penalty is minus 2. So in this case, the match score is +1, the mismatch score was -1, the gap was -2. So we just added -2, -4, -6, -8 on the first row and the first column after 0, right? Because accumulating gap penalties will be indicated by those horizontal and vertical jumps. And then what we had to do is that for calculating the score for each grid here, for example, this, we had to consider 3 paths. So one would be diagonal path. So this diagonal path basically represents this diagonal path represents a match or a mismatch, and the horizontal and the vertical paths, those will represent your gaps, right? Gaps meaning indels. Uh, and basically, after that, we had to consider this sort of algorithm. So the score for this particular grid ij, it will be the maximum of these 3 routes that we can take. So one was this diagonal route, match/mismatch. Another one was the vertical, and another one is the horizontal, which both represent the gap scores. And the max value among all of those would be considered for this grid. So here, for example, the max value was 1. And then similarly, if we just fill up the entire thing, so at the bottom right grid, whichever value we got. For example, in this case, it is 2 plus 2. So wait, I think I didn't share the screen. Sorry about that. Anyway, we just recapping stuff from earlier. Yeah, so what I was talking about is that the max value of these three routes, so diagonal route, horizontal route, and vertical route, those were considered for filling out a particular grid. And whatever was at the, let's say, bottom right corner, so that was our maximum or optimal alignment score. And then what we had to do is that for each time we filled up a grid, we just had to put an arrow. So basically tracing the path that we took to get to that grid. And ultimately what happened is that that gave us sort of the actual alignment, because we had to trace back from the optimal value. For example, in this case, uh, if we take the optimal alignment score of 2, so you have to trace back this path, for example, this path, this, this, and then this. And in this particular case, there was another alternate path. So we could have traced that also. And basically, like now we can consider, or now you can calculate the matches or mismatches, and also the gaps based on these diagonal or vertical routes. So basically, whenever we have match or mismatch, we just put everything at the same place. And whenever we have gap, for example, after CA and CA matched on the left and the upper side axis, so after that, we saw that there is this jump, right? Or there is this sort of indel. Now, in this particular indel, so this one I am talking about. So here, basically, uh, the upper side sequence, so that remained at the same position. However, the left hand side sequence that moved like one place forward. So from that we can just put a gap on the suitable place, right? And here we have since two different paths, kind of slightly different paths, so we could trace down that path to give us the optimal alignment uh, of these two sequences, right? So we discussed all of this. And then the Smith-Waterman alignment algorithm, which is for local alignment, what we had to do here is that again the process is same, but whenever we put, for example, a gap, or whenever we put a mismatch or gap or something like that, if that gave us a negative score, so what we had to do is that we had to put, instead of negative score, we had to put 0 in there, right? And then again, the process is the same. So we had to trace back the paths that we basically got that gave us non-zero scores. So for example, here the optimal alignment score or alignment score would have been +4, which is the maximum value inside this grid, right? So you consider all of the grids here, and then you had to just trace back, so which grid you got that value from. So this trace back would have given something like this. And then you would have gotten, okay, so GTGA from here and then this GTGA from here, they will be matching. So that is a local alignment, because you are aligning a smaller subsequence of this entire sequence, right? So that's fine. Another thing we considered, I just briefly touched in one of the questions. So that was conservation scores. Uh, so for conservation score calculations, there are some methods, but what Professor Grumiha mainly emphasized was this unweighted and entropy-based measure. So, can somebody in the audience say what is the importance of calculating conservation score for a particular position? So basically, the PAM matrices and Blossom matrices and so on, they give us like substitution matrices, like substitution probabilities uh, from one amino acid to another. However, they do not particularly show the conservation of particular amino acid at a certain position in the entire sequence. So they do not have position specific sort of parameters, so they do not indicate any position specific inference. So that is why these kind of conservation scores are important if you want to calculate the conservation of certain residues at a particular position. So here the formula was something like this. So summation of Fa times ln of Fa. And basically what we had to do is that, let's say in this particular case, uh, at the sixth position, let's say we had LAVKD. So all of these are different amino acids, obviously. And what we had to do is that for each of the amino acid that is present at this particular location or position, we had to calculate through this formula and then ultimately do the summation. So here what would happen is that out of these 5 sequences, because there are only 5 sequences, so leucine is present only one times, right? So you have to do 1 by 5 times ln 1 by 5. And then for alanine, again, the same thing, 1 by 5 times ln 1 by 5. And because every sequence in this one at position 5 is like occurring only once, so we had to do it 5 times. So 5 times 1 by 5 ln 1 by 5, which would give us a value of minus 1.61. So that is how we calculated conservation score. And we will talk about some of the position specific matrices. So that is not particularly for conservation, but what is the position of a particular amino acid to be like present at this point or at this location, just a little bit later on. So can you calculate this one? So here we have a question where the conservation score with unweighted and entropy based measure for position 9 is being asked. So can you calculate this one out? For position 9, that means for simplicity, if I just color code it, so it would be this position. So in contrast to the previous example that we saw, so here every amino acid at this position is not occurring once throughout this entire stretch of 9 sequences, right? Basically, we see that threonine is occurring 1, 2, 3, 4, 5 times. However, alanine, glycine, and lysine are present only once. So for this one, what would be the score? Can you calculate and put in the chat? So basically, since threonine is present 5 times, so you have to do something like this: 5 by 8, because for position 9 we are calculating. So that is total of 8 sequences, right? So for threonine, 5 time that is present at position 9. So you have to do 5 by 8 times ln 5 by 8. And then for alanine, glycine, and lysine, we do just 1 by 8 ln 1 by 8, 1 by 8 ln 1 by 8, 1 by 8 ln 1 by 8. So in total, it will give us, if we calculate it out, something like this: minus 1.073 or minus 1.07 if we take up to two decimal places, right? So that is the way to calculate sort of the conservation score, which can give us insight into the conserved nature at a particular location, right? Now, uh, so some of these simple sort of numeric kind of questions can appear in the weekly assignments as well as in the exams. So for example, here we have been given this sort of short stretch of amino acid sequence, right? LEADER. And then we have been given all of the molecular weight values for the amino acids. And then we have been asked, okay, so what would be the weight of the peptide that is formed by this sequence? So let's just take a moment to calculate this out, and you can answer in the chat also after calculating. So you have been asked that the weight of the peptide LEADER, and we have been provided the values for the individual uh, amino acids molecular weights. So Is it just that we have to add up all of these values for each of these amino acids, or is there something more? So please try to calculate it out and answer in the chat. So here, if you add up all of the molecular weights, so you notice that uh, E is present two times, right? Glutamic acid is present two times, and each other amino acid, those are present only one times. For example, L is one time, A is one time, D is one time, and R is one time, and E is present two times, right? So of course we have to do one times molecular weight of L, and then plus two times molecular weight of E, plus one times molecular weight of alanine, and so on. But this will give you a value of 810, which is also present here. You see, right? Now, is that the answer, or do we have to consider something more? So, does anything happen when, let's say, two amino acids are joined together? If two amino acids are joined together, what other thing happens? So, basically, what happens is that one water molecule is eliminated, right? So, in this case, not only do we have to add up all of the values for the molecular weights for the individual components, but also you have to subtract how many water molecules are eliminated during the formation of this peptide sequence. So since excuse me, since here we have six amino acids, so 6 minus 1 or 5 water molecules are expected to be eliminated, and that will mean that 810 from 810 we have to subtract 5 times 18. So if you do that, you will get a result of 720. You get the answer. So you have to just be careful that always it's probably uh, just simple summation will not give you the answer. So you have to consider what else happens during formation of the sequences and so on. So here water molecule is eliminated, and you have to eliminate, or you have to subtract the molecular weight of water from the answer. How many water molecules are eliminated? You have to count for that. Okay. So here at the answer will be answer D. So these kinds of questions you might face in your assignments, weekly assignments, and exams also. Now, let's say if we ask a question that you have to identify an amphipathic beta strand for a given sequence. If any sort of stretch of sequences that can indicate that, okay, it might be an amphipathic beta strand from a given sequences, then how do you identify that? So here the question is basically asking that. So try to again answer in the chat, and then we'll look at it. Okay. So first you have to consider, okay, what does empathic beta strand even mean? So you just consider that, and then uh, you can get to the answer. So can anybody mention in the chat again what does an amphipathic beta strand mean? So what is this amphipathic nature, or this amphipathic term that we are talking about? It is of course a characteristic pattern, you can say, uh, of a sequence where you are, based of some sort of uh, fundamental or basic characteristics of the amino acids, you are finding a pattern, and that pattern can indicate whether, like, what type of sequence or what type of features this sequence has. So one of those patterns, you can say, would be uh, the amphipathicity, I guess you can say like that. So try to answer in the chat and we'll look at it. So what basically amphipathic means, uh, that it is a pattern of polarity and then non-polarity and then polarity and then non-polarity, this sort of arrangement of the amino acid that are constituting this sort of short stretch of subsequence, you can say. So you have to look for a particular region where one polar amino acid is present after one non-polar, and then after then again just adjacent to that polar amino acid another non-polar is present, and then after that non-polar amino acid another polar amino acid is present, and so on. So you can refer to this chart on the right. So if you remember, in some of the earlier classes we talked about this term. This is a hydropathy index term, and also for simplicity's sake, here we also have the groupings of these particular sort of residues, amino acids, sorry. And then the aromatics, you can just consider their hydropathy index, and you can kind of get an idea from that. So this hydropathy index, what it was is that it indicated basically the polarity or the sorry, basically the polarity or hydrophilicity and then hydrophobicity of a particular amino acid, right? So this free energy of moving of one amino acid from a polar non-polar solvent to water, it was something like that, right? So here, if you sort of take a look at it, for example, the charged residues lysine, histidine, arginine, they have negative values, right? So these are negative values. So negative values in this case indicate that they are highly polar or charged. And then conversely, if you look at like valine or leucine or then isoleucine and these kind of things, so you'll see that they have a positive value. So positive values indicate basically hydrophobic amino acids. And for amphipathic beta strand, you have to kind of take a look at, okay, so one uh, one hydrophobic amino acid will be followed by another polar or polar charged or polar uncharged amino acid, and then that will be followed by again another non-polar, and that will be followed by again another polar, and so on. So if you look at these sequences, so if you, for example, consider these ones, so alanine, what is alanine? Here, of course, you see that alanine is a non-polar alanine has a non-polar R group, right? It has a hydropathy index of 1.8. Alanine, after that, you see valine. So valine is also non-polar, as you see here. And then two alanines, and the threonine also is there. So threonine has slightly, negative uh, value of the hydropathy index, but it is a polar group. However, these four residues that are preceding threonine, those are all basically non-polar amino acids, right? So there is no such periodicity, you can say, present here. Similarly, in this one also you will see no such periodicity of polarity. Polar amino acids and non-polar amino acids are present. Here again, two states of non-polar followed by a charged amino acid and another non-polar amino acid is present. The most appropriate in this case would be this one here. The answer A, if you notice, you have an alanine which is non-polar. After that you have lysine, charged polar, right? And then you have valine, again non-polar. Then you have threonine. Threonine is again polar, polar uncharged. Here it is mentioned. And then after that alanine again, which is non-polar. So basically this stretch uh, can be considered an amphipathic, right? So in this case, the appropriate answer would be answer A. So you have to, I guess, try to remember the classification of the amino acids based on their polarity. So the non-polar and then polar charged, or uncharged polar, and so on. And then what the aromatic R group containing amino acids in what classes those fall into. So you have to remember that, and then if questions arise which asks for a pattern of polarity and non-polarity, then you can sort of use that to get to the answer. In this case, the answer was answer A. Now, on the concept of amphipathicity, here we have, of course, some examples, right? So, for example, in this figure A, so the figure legends are, I guess, given on the right side. So that is the alpha helix of PEX11, which has a basic polar phase and a prominent hydrophobic phase. So in this case, you notice that the upper side, basically where these blue and red sort of cylinders are indicated, those are indicating the amino acids right here. For example, blues are indicating arginine, lysine, histidine, and then the red cylinder is indicating aspartate and glutamate. Of course, these are charged amino acids, polar amino acids, right? So those are present on one phase, and then on the other phase, if you notice, you will notice uh, this aromatic group containing amino acids and these non-polar amino acids, right? These ones for yellow. So you see that basically on two faces, so the upward and the downward face, or the upper and the bottom face, you see basically two types of amino acids. So the periodicity or the amphipathic nature of the sequence, that is sort of creating this kind of pattern. Uh, and then if you notice for the other ones as well, so you'll notice similar things, right? So A, B, C, D, E, F, all of the examples in this one, they have this sort of particular arrangement for amphipathic amino acids. And basically, this figure 2, so what it mentions is that uh, for amphipathic nature, some sort of particular pattern is observed. So here, I plus 3 and I plus 4. So I, if you consider the start of a hydrophobic amino acid, so the next hydrophobic amino acid for helices, that will appear at I plus 3 or I plus 4 position. For example, in figure B, if you notice, so you'll notice that if you start at this gray position 1, which is a hydrophobic amino acid, let's say, so if you jump from 1 to 4, it will be, of course, I plus 3, right? And then after that, you jump from 4 to 8. So this 4 to 8 jump will be I plus 4, because 4 plus 4 is 8. And then again from 8 to 11, you see I plus 3 is indicating the new amino acid. 8 plus 3 equals 11. So basically here also we see sort of a periodic nature of this I plus 3 and I plus 4 pattern. So from first to second, you had to jump plus 3. Second to third, you had to jump plus 4. And then the next one, you have to again jump plus 3. So these sort of patterns might be possible, visible in basically amphipathic states of helices. Okay. So here are some other examples, as you saw, heat shock proteins and so on. So these are also important proteins. And so by just analyzing the sequence, you can get one idea from here. That just by analyzing the sequence, what sort of pattern you can get from there, you can basically understand, or you can basically get insight into some sort of features, for example, amphipathicity in this case, right? So that's quite interesting. So the references are given here below at this slide. So we can just quickly look at how this sort of pattern is important in the functional context, right? So first of all, for example, here, so for text 11, which we also talked in earlier slide. So here, a sort of membrane deformation is induced by the alpha, by the age or the amphipathic helix of the peroxisomal membrane protein PEX11. So if you notice here, as we saw in the earlier slide also, PEX11 had basically separation in the two sort of phases, right? Upper phase and lower phase. One was your polar, and then another one was non-polar phase. So here you saw that this sort of stretch of amphipathic helix, it is basically being injected or introduced inside this membrane. And what happens is that it sort of creates a membrane deformity at this position, right? And that helps in certain functioning of this, of this cellular membrane level context, right? Then again, we are seeing for B, for example, guanosine triphosphate GTP/GTP exchange in ARF1 that controls the exposure of its amphipathic helix and thereby translocation of the small G protein to lipid membranes. So again, here you see that the for the ARF1, I guess that is also carrying this GDP, then helping, I guess, its translocation. So that is again done by introducing the amphipathic helix into the membrane itself. Heat shock protein we know very well. So heat shock proteins in this case are also, uh, we see amphipathic helix's relevance in this case. So here the adsorption of large amphipathic helix region of the heat shock protein HSP12 in figure E. So that has a protective effect on the plasma membrane. So because while we introduce this amphipathic helix into the lipid membrane, so that sort of changes the characteristic of the membrane. And here in this case, by adjusting the physical properties, it has some protective effect as we see here. So this amphipathic stretch of residues are inserted into the lipid membrane here, right? And another very interesting example, which I think was here somewhere. Yeah. So for the example, if you are seeing that the amphipathic helix of PLIN4 that coats lipid droplets under conditions of insufficient phospholipids by directly substituting the phospholipid monolayer. So if we sort of look at this position, so here this PLIN4, whenever there is insufficient phospholipid, so let's say phospholipids by some condition is not being produced at the correct amount. So what it does is that it introduces this PLIN4, it introduces the amphipathic helices into the lipid membrane, and it substitutes for the phospholipid monolayer. So that's quite an interesting mechanism by which certain functionalities or structural properties are being adjusted for. So those are some examples taken from this particular source. Okay. Again, just another small example was like beta barrel proteins. So of course, the beta barrel proteins will have two faces. So one that is facing the helices, and then one at the core position. And again, though that would indicate amphipathic nature. Okay. So let's say we have to calculate the weight matrix value for alignments, right? So here in this case, the question is asking for the DNA sequences that are given below, what will be the value of T at position 2 in a weight matrix. So this is not just an alignment matrix, but this is a weighted alignment matrix or a weight matrix. So position 2 will of course indicate this uh, this position, right? A, G, G, G. So what would be the value of 2 for this weight matrix at position 2? Sorry, what would be the value of threonine for this weight matrix? Uh, So could you please try to calculate the answer and share in the chat, or you can unmute also. Just try to calculate it out and share in the chat. So firstly, you have to construct an alignment matrix. Okay. So what an alignment matrix does is that, for example, on the left-hand side, we have all of the individual components. And on the upper side, here we have the positions. So this will be the positions, and these are the bases, or you can say individual components, right? So first you have to calculate the alignment matrix, and then you have to apply the formula for the weights. Just try to calculate it out. So how do you exactly fill this alignment matrix? So can somebody mention what code will we put here? So basically this codes will indicate that out of all these sequences, for example, 4 sequences are here. For position 1, how many alanines are present overall? So at position 1, how many alanines are present across these 4 sequences? So yeah, 3 by 4, but in this particular case, we'll just consider the exact count. Okay. So let's say 3 alanines are present at position 1. Similarly, you could just see, for example, at position 1, no C is present. So that will put we will put a 0 there. Then G is present 1 time, and the T again is present 0 times, right? So by doing that, we will just fill out this entire matrix. So for example, if I ask what will be the score at this particular grid, so that is for position 4, G. So G for position 4. Yeah, I see in the chat 0. So basically 0, right? And then if you fill out this entire matrix, so that is how you get the alignment matrix. Now you have to apply the formula for the weight, right? So what weight matrix will do is that it will take the alignment score for this, the score for this particular grids, and then you have to apply some additional formula. So again, if you put it like this, so basically what this formula is saying is that this pi, so this term right here, so this pi will be the probability for having one of the particular components at this position. So in this case, let's say if we talk about this grid, so that will mean the probability of having alanine at position 1. Now, what would be the probability of having, sorry, not alanine, this is, adenine. So the probability of having A at position 1. So in this case, since only 4 nucleotides are present, right? We only have ATGC for your DNA sequences. So this probability will essentially be 0.25, because if you divide 1 by 4, so the total probability is 1, and any of the 4 nucleotides can be present here. So you can just say 0.25. If it was amino acid sequences, you have to divide by 20 for naturally occurring amino acids, right? So that will be 0.25. Now this Nij, what is that? That is the number that we got for this particular grid. For example, here 3 we got for this particular grid, and N is the number of sequences in total. So here we had 4 sequences, right? So that would mean n by 1, n plus 1 will be 5. So could you calculate the score for the first grid here, the grid that I'm highlighting here? It will be Ln Nij plus Pi divided by n plus 1, which will be again divided by Pi, and you have to take the logarithm, natural logarithm of the entire thing. So if you try to calculate, for example, the first grid, so the inside value, what it will be. So basically, since Nij was 3 plus Pi, so for alanine, in this case, we got 0.25. So that will basically mean 3.25, right? So 3.25, again, divide that by n plus 1. So since it was 4, so 4 plus 1 would be 5. Uh, so that will be 3.25 by 5. And then you divide that entire thing by Pi. So again, 0.25, right? So then what will be the score? You have to take the natural logarithm of the entire thing. So if you just calculate it out, you will see something like 0.955, which should give you, 0.96. And for example, if you, fill out the entire matrix, it will just, follow that rule only. So let's say we want to calculate this one. Can somebody give the answer? So here your score is, or grid value is 1. So then what will be the answer? So it will be, yeah, I see ln of 1, 0. Yeah, basically. So it will be basically 1.25 divided by 5 divided by 0.25, something like that, right? So basically 0.25 divided by 0.25 equals to 1. So ln1 will be 0. So like that, you have to calculate for each grid. And then here the question was asking for position 2, threonine for position 2. So threonine for position 2 is basically this one. So minus 1.61. So that will give the answer of minus 1.61. So if you had asked the weight matrix values, for a given sequence alignment, you have to just calculate it out like that. Okay. So this question was, I guess, in one of the earlier assignments. Assignment for one of the earlier iteration of the courses. So these kind of questions might come in the assignments also. Okay. Now we talked about the concepts. I guess Professor Gumi have mentioned uh, the concepts of this matrix, which is called PSSM. Does anybody remember what it stands for? Yeah, so I see one answer. Position specific scoring. Yes, position specific scoring matrix, right? So what does position specific scoring matrix indicate? So again, uh, we are constructing just yet another matrix. It will be scoring for specific positions in alignment sequence, right? So what happens is that it is basically, like a table containing probability information about amino acids or nucleotides at each position in an uncapped multiple sequence alignment. So in this case, the multiple sequence alignment that is considered, that will be ungapped. So ungapped is considered because it will be fixed length alignment kind of. If you would introduce variable length, sorry, gaps, then it would be variable length sort of an alignment, which will make this coding bit kind of difficult. Uh, and again, the focus here is to identify, for example, conserved set of subsequences, right? So here you will only like use this for very closely related sequences where there is not that much difference. So the gaps or indels, which are basically only happening like throughout a higher evolutionary distance and so on. So the gaps of sequence do not, again, they are unfavorable, so they do not occur that much. So basically ungapped multiple sequence alignment is considered in this PSM matrix. And what you would have noticed is that uh, it basically indicates positional information, just similar to our earlier matrix alignment and weight matrices that we considered. And here you can, let's say with rows, you can specify the residue positions, and then the columns, you can specify the residue names or the characters that you are trying to construct for. And then you can also do the other way around. And basically, you have to again convert these to log odds scores. So similar to our earlier TAM and Blossom matrices and so on, we converted the actual scores to some log score, so log based score. So here you have to again do the same thing. And then for each column, you have to count the raw frequencies. Then you have to do normalization to ensure the length and composition independence. And then log base of 2, you convert that. And basically, positive score in the matrix will indicate identical or similar residue matches, and negative score will indicate non-conserved sequence matches. So positive and negative, two types of score you can expect to get. Right. Yeah, basically it will give a quantitative measure of sequences conservation at each point. So how does this Poisson specific scoring matrix work? So here, first of all, you have to convert the multiple sequence alignment to I guess a raw frequency sort of table. So let's say we have to calculate the frequency of alanine occurring at position 1. So what would be the value? Can somebody mention in the chat? So we have, for the 5 sequences that are given here, we have 1, 2, 3, 4, 5, 6 positions. So if you are asked what will be the frequency for occurrence of alanine, frequency of occurrence for alanine at position 1. So it's very simple. So earlier we used the raw counts for each position. So how many alanines was present? Earlier we considered that. Here we just want a frequency, right? So a total of 5 sequences are present, and out of that, if we notice, we will notice 3 alanines are there, right? So it will be 3 by 5. I think. Yeah, I also see answers in the chat. 3 by 5. So 3 by 5 is basically 6 by 10 or 0.6, right? And then you have to just fill out the entire matrix right there. For example, if you are asked like at position 1 threonine, what would be the score? So you just count. Okay, so there is only 1 threonine at position 1. So it will be basically be 1 by 5, right? 1 by 5 equals to 0.2, 2 by 10, 0.2. Similarly for, well, if you consider this grade, so at position 4, the occurrence of threonine. So at position 4, we see 1, 2, I guess threonines are present there. So that will mean again 2 by 5. So 4 by 10. So 4 by 10 is 0.4. Right. So that's how you can calculate the frequency of occurrence for a particular position. Now on the right side, what you also have, if you notice here, you have something called overall. So this is basically the overall frequency in this entire alignment for a particular, let's say, nucleotide here, right? So for a particular nucleotide, the overall frequency has to be considered. So if you are, let's say, counting for alanine, how many alanines are present in the overall alignment? So irrespective of their positions, how many of them are present. For example, we have one alanine here, right? And then 2, 3, 4, 5, 6, 7, 8, 9. So total 9 alanines we are seeing. And in this total set of sequence alignment that we have, total 30 residues are present, right? 5 times 6 equals 30. So if you divide 9 by 30, so it will give you 0.3. That's how you can give sorry for the that's how you can give for adenine, right? You can get the value for adenine. I guess I kept saying alanine there. Here we are only considering nucleotide, so it will be adenine, right? So the overall for A will be 0.3, and then for T, I guess it will be 0.2, 0.27, 0.23 if you just count the overall number of A being present in this entire sequence alignment. So you'll get the frequency for that in this order, right? 0.3, 0.2, 0.27, 0.23 A for ATGC. And then what you have to do is you have to normalize the values by dividing them uh, by their overall frequency. So normalize the values in the sense that in this range, so the actual position specific scores that you have to normalize that. And what are you going to normalize with? With this overall value. So this overall value on the right side, right? And if you do that, you will get something like this. So 0.6 divided by 0.3 will be 2, and so on, right? So for example, here for threonine, 0.2 divided by 0.2, that will be 1. So here you have 1, and so on. So you have to just divide the value for each grid with the overall frequency of that nucleotide here. And then again, as for all of these matrices, we convert to basically log to the base of something. So here we are converting to log to the base of 2, and then that will give you sort of this kind of matrix. Okay. And again, if you are basically, if you notice, so for the sequences for the nucleotides at particular positions which are present like a lot more times than the others, let's say for example here alanine. So here we saw in the raw frequency it was 0.6. So we are also getting a positive value for adenine in this particular one, right? And then that's how you can basically interpret this entire matrix. So for in a particular grid, if no element is present there, so for a particular position, if let's say C or G was not present there, so you just omit those values. And that is how basically you construct a position specific scoring matrix. So yeah, that's the concept. Now a lot of these matrices, what you do is that you calculate the log based score, right? So the reason that we calculate the log based score is basically that. So first of all, what does log based score do? So if you are, for example, calculating probabilities and so on, so sometimes the probability for a specific thing could be very low. Let's say you have 0.0006 or something like that. That sort of probability, right? Let's say the probability was 10 to the power minus 4. So if you are calculating certain things using 10 to the power minus 4, it will be very difficult because you will get very small basically a value. And then again, if you're using some algorithms, you could get into like the floating number imprecision and so on. So basically what it would do is that if you convert that to logs, for example, so 10 to the power minus 6 log, you will get minus 6, right? So basically like that. So it will convert to a more manageable sort of set of scoring for each of the grids. So I was talking about log base 10. And then again, it sort of narrows down the ranges, which for a lot of algorithms are very good because they can calculate at a smaller range. So the smaller range will sometimes improve the accuracy of the algorithms. And again, for the log conversion formulas for multiplications and so on. So if you have log of let's say a times b, you can just convert to log a plus log b. So that again streamlines the basically computational cost, you can say, in a way. So that will be sort of faster in some way. So those are the reasons why in a lot of these cases you will see that log-based scores are included in the matrices. Okay, so that was basically the concept of position-specific scoring matrix or PSSM. So the examples are taken from this book right here, which I mentioned at the bottom. Okay, So here we have again another question that asks that if you plot the hydrophobicity indices, let's say the measurement of how hydrophobic or how polar they are, the residues, amino acid residues, against the sequence number. So what is the term that the result is called? So can somebody mention in the chat? So hydrophobicity indices you can kind of think of in a similar way we talked about, about the hydropathy index. So kind of similar thing, just a measurement of hydrophobicity using some sort of uh, parameters, right? So in this case, what you will get is basically it will be called a profile or a profile of hydrophobicity, a hydrophobicity profile. So it could look something like this. For example, if you have in this plot, so let's say on this axis you have basically the sequences, and in this axis you have some values for the hydrophobicity. Let's say H value or something like that, if you call it. Then what you will see is that you could see some sort of patterns, right? Maybe something like this. So some sort of stretch of sequences, residues that will have high hydrophobicity, some will have low hydrophobicity on the negative value, and so on. So this could give you a sort of idea about what is the pattern or the arrangement of hydrophobic amino acids and polar amino acids in the sequence. So if I ask one question, uh, let's say we have a hydrophobicity profile which looks something like this. Let's say uh, like something like this. Okay. So a lot of sequences are considered, let's say here. So this, let's say, is a hydrophobicity profile. So how can we smoothen the hydrophobicity graph here? How can we smoothen the plot? Smoothen the plot, make it look something like this one. How can you do that? So if you remember some of the earlier uh, content discussion that we had, or from Professor Gomia's classes. Uh, so in the cases of, for example, visualization matrices like dot matrices and so on. So if we considered uh, for a long stretch of sequences, each character by character match and put a dot there or not. So you might remember that it looked like lot of dots were there, right? So it's very much like a noisy sort of plot. Sometimes it's possible that we cannot infer anything of significance from those kinds of plots, where it is like filled with dots all over the place, and then you cannot find a specific diagonal pattern that you are looking for. However, there was one particular way that we could reduce the number of dots, if you remember, in those cases. So the same thing can be applied here. So basically, the concept that we talked about in that context, for the dot plot context, we introduced the ideas of window sizes or a sliding window technique, right? So what we did is that we, instead of matching one character against another character, we used a window size of a particular value, for example, window size of 3, and then if based on our requirements of stringency, like how stringent we want the match to be, so based on that, we put a dot or not based on a window size of 3 basically. So obviously that will reduce the number of dots, in comparison to 1 by 1 matches. So 3 by 3 matches will have a lot lesser number of dots. In this case, you can do the same thing. You can like have some window size, and then you can calculate the average, basically value of hydrophobicity for that window, and then you can sort of smoothen the graph to look a bit more like this to kind of infer some sort of patterns, right? So that you can do basically. Okay, so we talked about normalizations as well. So in one of our earlier examples and the position specific scoring matrices and so on, we used normalized scores, right? So in a lot of cases, we have to, because if you have a large range, it would be like kind of hard uh, to just visually understand, or just on the go understand what is going on, like what is the pattern going on, what does a particular value indicate. In a lot of those cases, what people do is that they normalize the entire set of value to be present between a certain range. So here in this case, the question is asking that if you have these numbers 2, 5, 10, 15, 20, uh, you have to normalize these numbers between 0 and 1. Okay, you have to normalize these numbers between 0 and 1. So how to do that? So can somebody try to do it and answer in the chat? So normalize the entire stretch of values between 0 and 1. So the minimum value should be 0 and the maximum value should be 1. And then the middle values will also be transformed to be something between 0 and 1. So the formula for normalization looks something like this. It will be x minus the minimum value. So x is one particular component. So if you say 2, 5, 10, 15, 20, whatever. So x minus the minimum value divided by maximum minus the minimum value. So using this formula, you can normalize, let's say, the values uh, between the minimum and maximum range. Between 0 and 1 in this case, right? If you notice some sort of pattern here, for example, so let's try to do the first element here. So 2, for 2, what it will be? So 2 minus the minimum value. Minimum value in this case is 2, right? If you compare 2, 5, 10, 15, 20. So 2 minus 2 divided by max minus min. So max is 20 divided by 2. So that will be 18. So 2 minus 2 is 0, right? So then this value will be 0. Similarly, if you take a look at the maximum value 20, so what it will be? X in this case is 20, x minus min, and then max minus min again is the same thing. So it will just cross out, right? It will be 1. So now you see, you have the lowest value as 0, the highest value as 1, and then in the middle values, those will have some sort of values transformed in between the range, right? The range of 0 and 1. So what would be the score for 5? For 5, it's 0.17. I see in the chat. So let's just see. So for 5 again, if you try to calculate, so 5 minus 2 will be 3, then max minus 2 will be 20 minus 2. So basically 3 by 18, right? It will be 0.166, something like that. So 0.167, or if you take up to 2 decimals, so 0.17. Yeah, 0.17 up to decimal, right? So it will be something like that, right? 0, 0.17, 0.44, 0.72, and 1. And here you have the normalized set of scores. So using this formula, you might remember this one. So if you are asked to normalize between 0 and 1, so you can use this formula. Okay. Fine. So if you are asked phylogenetic analysis for protein sequences, that gives information about what? So Professor Gromiha's classes, Professor Gromiha went over a lot of concepts regarding phylogeny, right? So what are the phylogenetic analysis meant for? Could you try to answer in the chat? Okay, yeah. So I see one answer in the chat B. So phylogenetic analysis for protein sequences is essentially done for evolutionary relationships, right? Okay. So we'll just look at in some further slides about some sort of calculations, you can say, how these phylogenetic trees are sort of derived. So we'll look at some examples just in some of the further slides. So before that, let's say you are asked for hydrophobicity profile, what sort of window size will give you the smoothest plot. So we just discussed this earlier. We can smoothen a hydrophobicity profile by allowing for a window size, accounting for a window size. So the window size will mean, let's say we just take a long stretch of subsequences and we just count that as one. So we have to calculate something like the average value, let's say, and then we can put it corresponding to that one, right? Corresponding to that stretch of subsequence. So if you introduce window size, of course the plots will be smoothened. And which window size will give you the the smoothest plot here? So here the answer actually will be the largest window size will give you the smoothest plot. So the smoothest mean more number of noise will be reduced. Okay, I mean number of noises will be reduced, but the smoothest will mean that uh, you can get like the maximum reduced noise. Of course, you cannot increase it as much as you want because that will basically be sometimes be neglecting the entire uh, sort of patterns or sort of set of values that you can get for these sequences. It can sometimes prevent you from inferring any significant meaning also. So you have to be careful while choosing window sizes. Uh, but let's say if we have a long stretch of sequences, let's say 2000, 3000, something like that. So there, of course, you can use, let's say, large window like 10 or 15, 20, something like that. If you only had, let's say, 100 sequences, then if you are already introducing like 10 or 20 window size, so you will not have that much data points to put on this plot, and that will not allow you to infer any sort of patterns. But for larger stretch of sequences, it's okay. So you can try to uh, manipulate the window size and try to see which window size gives you the best information regarding the patterns. So the point here is that you are trying to observe some sort of patterns uh, in the values for the hydrophobic indexes here, right? So here, for example, just to recap the concept of dot plot, what we had is that character by character matching would be something like this. So that's fine. However, if you can use a larger window size, so you have to basically slide the windows. So for example, ACG versus ACA with a stringency of 2, meaning that exactly if two character or more matches, then we can put a dot there. And here ACG versus ACA, two characters are matching for the first and second positions, so A and C. So you can particularly put a dot here, right? Similarly, you have to, in this case, we used like overlapping sliding, right? Overlapping sliding means that after ACG, the next window, let's say you can count is CGC. Yeah, so after ACG, the next window you have to count, like for example, CGC, these three things, these three elements, right? Here again, you are finding like matches for CGC and CAC here. So you can put a dot here because C and C on the first and third position matches. And then you can fill out the entire matrix. And then you will notice in the end, the number of dots is obviously reduced from the initial basically grid with one by one match, one by one, I mean, 1-to-1 match, right? Here. So the number of dots are reduced. The number of noise will be reduced in some sense, and the plot, if you are like plotting it in the case of hydrophobicity index, that will smoothen the plot. Okay, so we were introduced to the idea of phylogenetic tree constructions, and the phylogenetic tree constructions, for example, Professor Gomiha like talked about some of the concepts or some of the processes that you can implement. One of those was UPGMA. So this is unweighted pair group method using arithmetic mean, the full form. Some other concepts, I guess, like Raymer joining, maximum likelihood, etc. Those were also mentioned, but mainly focusing on UPGMA. So this is basically a distance. So this is basically a distance-based approach where you sort of try to calculate the distance between two sequences. And how are you calculating the distance between two sequences? You are accounting for the mismatches that occur there. For example, let's say we have two sequences, right? So let's say we have amino acid sequences A, T, A, L, K, M, A, B, Y, something like that. That is sequence one. Let's say another sequence that you have where we had, uh, let's say A, T, V, L, K, um, let's say A stays here only, let's say A, Y, right? So if you compare these two sequences, just counting the number of mismatches. So what are the number of mismatches that are occurring here between these two sequences? So these two look pretty similar. However, some difference is there for certain positions, right? So if you count the total number of mismatches, what will that be? I see one answer 2 in the chat. Uh, the others can try also. Basically 2 will be the answer because we have a mismatch at this position, then you have a mismatch at this position, right? And the rest are matching kind of. So two sequence, uh, two position mismatches we are getting here. So you can sort of think of it like that. That okay, the distance between sorry, the distance between these two sequences is 2, let's say, right? So we are just accounting for the raw values here, 2. Now let's say one more sequence comes into the picture. Let's say it is called sequence 3, and let's try to just construct it, right? Uh, so we are, let's say, comparing with 1 as our difference sequence, right? So let's do it. Okay, so let's see this one, right? 1 versus 3. So how many mismatches we are getting between 1 versus 3? So the last one is F, okay, phenylalanine. How many distance, or what is the distance between 1 and 3? How many mismatches we are getting here? So I see one answer 4 in the chat. Others can also try. So if you compare, uh, you have ATA, these are matching, of course. Then at the fourth position, you have P. This should be F, I guess. Or you can say P, I guess. That's fine also. So let's say this one is also mismatching. So here you put just a cross here, 1. Then L versus K is another mismatch. Oh no, that was with comparing one. If you are comparing with one, then yeah, K versus K, that is match. And then M versus V, that's a cross right there. Then again, you have at the last two positions. So V versus T and Y versus F, those are crossed. So that is a match. So here you have total of 4 mismatches, right? So you can say, let's say the distance between 1 and 3 is 4, the sequence 1 and sequence 3. Now, if you are asked, okay, so between 2 and 3, 1 is closely related to which of those? So between 2 and 3, sequence 2 and sequence 3, 1 is more closely related to which of these two? If you are asked that, then what would be the answer? Try to answer in the chat. So I see sequence. Yeah, sequence 1 is more related to sequence 2. So is that correct? Others can also try to answer. So 2 and 3, which one is more closely related to 1? 2. Yeah, so 2 will be more closely related to 1 because it has more similarity with 1, right? Or you can say it has less distance from 1 if you are considering distance as the number of mismatches that we get here. And since 3 has a higher number of mismatches or a higher distance from 1, so from this whole concept, we can see, okay, 1 and 2 are more closely related than 3. And that is the concept of constructing the phylogenetic trees, where let's say each of the sequences that you have will be considered as a node. And what you have to do, you have to just figure out a way to calculate the distances between all of them, between let's say pairs and so on. And then you have to see, okay, so if I see these two distances are very small as compared to the others, then those will be closely related. Then these other two set of sequences, those are closely related. These two seem to be very far apart, so there are a lot of mismatches occurring here. So those will be further down the evolutionary divergence, evolutionary timeline and so on. So that will give you an idea of how closely or how far through an evolutionary process the sequences are present, and that is kind of the underlying idea here. So here for the UPGMA method, so it is basically a clustering method that is mentioned here that constructs a tree-like structure by accounting for the sequences, and it is a sequences. It is a sequential clustering approach. So you have to sort of do it one after another, and just we will talk about how it is done. Uh, so it starts by identifying the two taxa with the smallest pairwise distance in a given distance matrix. Now we talked about distance matrices. Let's say we talked about alignment matrices earlier. So let's say it's kind of the same thing. For the sequences, you just construct a distance matrix. So what is the distance between one set of sequence and the other set of sequence, or one sequence and the other sequence, right? and you just put those values, let's say some sort of normalized or log form values in a matrix form, and then in that matrix, whichever will be the smallest pairwise distance value. So the smallest pairwise distance value. So you can just firstly consider that, okay, so these two are sort of the most closely related among this entire matrix, right? And then after that, what you will have to do, so you have to place a node at the midpoint or at the half distance between these two taxa. So you are saying, okay, so these two are related, and let's say their common diverging point was this particular node. We are placing a node here, and this node will represent basically a new composite taxon. So what it basically means is that, let's say, sequence A and sequence B uh, were found to have the smallest pairwise distance value in the distance matrix. So we will just put a node in between them, and then we will consider A and B as one single unit. So the next step will be to reduce the matrix, to reduce the size of the matrix, where A and B are considered as one unit, and we are evaluating the distance of the other elements, let's say C, D, E, and so on, from this unit A and B, A, B together, right? So that's how this code will proceed. Scoring will proceed, and then let's say you are finding another sort of element C, forget C. Let's say you are finding another element D, right? So A, B, you are finding that from this unit AB, D is having in the reduced matrix kind of, D is having again the smallest pairwise distance. So in the next step, what you will do is that you will consider placing a node between this AB composite and the D, and you have to consider ABD total, this total unit as one single unit, and then you will proceed further. So that will give us like a path of how to construct the phylogenetic trees basically. Yeah, so it is a reduced matrix. A reduced matrix is then created by treating the new clusters as a single taxon, and you have to just iterate over this process one after another. Uh, Grouping processes continuing until all taxa are placed on the tree, and then you have to do some outgroup routing, etc. Some assumptions are mentioned here. So the UPGMA method assumes a constant rate of evolution, so that all taxa evolved as a constant rate and equally distanced from the root. So from the root, that means that, let's say, A and B, we talked about, let's say at the initial values, we are finding that A and B have the lowest sort of distance value on the matrix. So what we do is that we put a node in between them, and let's say we are saying that this A and B are diverging from this node, right? Here constant evolution rate is assumed. So that means that the distance from this node to A, so this distance and then this distance, we are assuming those to be equal. So that is the assumption here, constant distance from a particular root, or let's say diverging point. Yeah, so that kind of assumption is here. Limitations is that constant evolution rate can often deviate from the real data. So potential erroneous tree topologies can be obtained from that, and then it is using, it has its usage in DNA microanalysis and so on. So the information are taken from this book. We will see an example of how it is done exactly. So let's say this is some sort of distance matrix, some sort of normalized matrix, let's say. So we have four unique taxa or four species, A, B, C, and D. And we are comparing something, some protein, some, their genome and so on, something like that. We are comparing something, right? So A, B, C on the top side, we are mentioning here. And then BCD on this side, we are mentioning here. We are not putting ABCD versus ABCD because, again, diagonal will basically mean the same thing. Here, the size of the entire box is reduced a little bit. So it's basically ABC versus BCD. Uh, and then A versus B, you have a value of 0.4. Then A versus C, we have 0.35. Then 0.60 A versus D. B versus C and B versus D is 0.45, 0.70, and C versus D is 0.55. Right. That's fine. We are not repeating or we're not putting the values for the same sequences because there will be no distance between them, right? Okay. So what will be the first sort of nodes placed between which of these two or which of these four um, different unique taxa can we place the first node? Can somebody mention? So between which of these four taxa in the construction of phylogenetic tree can we place the first node, or we can use that as the first step? So if you remember, the lowest distance values will be the starting points for constructing these trees, right? So could you answer this one in the chat maybe? So here, if you look at this matrix, you will see that 0.35 seems to be the lowest value in the matrix, right, in this distance matrix. And 0.35, of course, corresponds to A versus C. So what you have to do first is that, okay, so that will be, so you will place the first node between A and C, like this, right? And let's say they have a common point here, and the branch length, what is considered? So the branch length will be considered as half of the entire value of this distance, right? So here, since it was 0.35, we have to divide it by 2. So 0.35 by 2 will be 0.175. So this branch you can kind of say 0.175, and then this length is also, but not that. So this particular length, so that is 0.135. Let's clear it up. This one is 0.175, 0.175, this one is. So after that, if you remember, in the construction method, we had to consider A and C in this case, because those are the lowest distance value. So A and C as one single unit, right? So what we will do is that in the next step, we will consider AC versus, let's say here B and D, and then here also we have B. So that will account for the entire set of um, species that we are present that are present here, or taxa that are present here. So AC versus B, now you have to calculate it again. So for AC versus B, what is done is that it calculates basically from the original box. It calculates as AB plus uh, BC divided by 2. Okay. So each of the component of this unit, basically A and C, what was their original distance from B, right? So that from A to B distance would be AB, or A versus B. From C to B distance will be C versus B. And in the original, if you see, A versus B was 0.40, and then your C versus B was 0.45. So basically what it will be is that 0.4 plus 0.45, divide that by total, so you average it out, and then you get a value of 0.425. So that will be your new value for this grid AC versus B. So B versus B is blank because it's the same thing. Again, AC versus D, what will that be? It will basically be from each of the components, what was the distance of B from them, right? So originally you had AD plus, let's say, CD divided by 2. So here again, AD was 0.60, BD was 0.70, or is it? Wait, AD plus, AD plus CD, sorry. AD was 0.60 and then CD was 0.55. So 0.55 plus 0.6 divided by 2, that is 0.575. And B versus D, again, it was 0.7 as mentioned. Now in this new grid, what is again the lowest value? So here we are seeing, of course, 0.425 is the lowest value in this grid, right? So what we have to do again in this next step is that B and AC, this unit we have to combine, right? So in this new step, okay, firstly, before that we have to put a node between AC as a single unit and B, right? So here it can be done as this way. So AC, this sort of uh, the node between A and C, that was in our first step only, and then here you are introducing B, which will have a common point with the entire unit A and C combined, right? Uh, and in this case, the basically distance from this common point, let's say, to B, that will be calculated by 0.425 divided by 2 equals 0.212. That's how the distance is calculated here. Half the distance is the total value is halved, and then you can think of it as the distance of B from the common point, right? Now, in the next step, again, what you have to do, as I said, you have to combine B with AC, right? So you have to do something like this: BAC, and what will be the value of BAC with D? Like, what is the value here? So the value, of course, would be individual components against B in the original box or the original grid. So B versus D, BD plus AD plus AC divided by, since three of them are here, so 3. So if we see here, BD was 0.70, so that's here, and then AD was 0.60, so that's here, and then CD, that's 0.55, that's here, and then add that up, add all of these up, and then divide by 3, you get 0.617. So yeah, so basically what you are doing is that, let me just clear some of this stuff. Yeah, so what you're basically doing is that, again, the combined set of nodes here, for example, A, C, and then B all connected, you are just putting D uh, as originating from a common point between all of these, right? So here, so this common point to D distance again will be calculated by this value divided by 2, because you are halving, basically you are making the total value half, so 0.309. So that's sort of a simple construction of a phylogenetic tree. And this again represents that, of course, A and C, they are very closely related, right? The most closely related among these four. And then it seems that A and C have a common ancestry with B, which is starting at this branch or this sort of common point. And then this entire unit has a common originating point from common originating point with D. So that sort of stepwise way you can relate which one of these are more closely related, which one of these are present at a higher evolutionary distance, and so on, right? Okay. Mm, so in the uh, phylogenetic tree construction, so after constructing that, so the trees are basically represented by some of the notations. So here the Newick notation is mentioned, and let's say you have this constructed tree. So here, of course, you are seeing A, B are together, and then it seems to have a point here, common point here somewhere, like D and E are together. Those two units are having a common point with C, and then C is having common point with A, and so on, right? So if you convert that to a Newick notation, uh, what will be the notation for the Newick method? I'll try to answer in the chat. We will see. I see one answer C. Yeah, two answer C. So basically, how you construct is that you whenever you see two nodes that are connected, you just group them together using a parenthesis, right? So for example, here this A node and then B node, these are connected from a common point, right? Through a common point. So what you will do is that firstly you will just group them together and then just put a parenthesis around them. Now another observation here, you can see that D and E are connected through a common point. So let's say we just put D and E and then we just put a parenthesis around them. And with this unit, so this D and E is common point, we see another node which is C that is connected right here. So you have to put C here and then again you have to group this entire thing together because C is connected with as a whole D and E, right? The common point of D and E between those. And then F seems to have uh, the point here which is connecting with the A and B unit as a together and then the CDE unit as together. So yeah, you can just put a comma here, then put a comma here, you do F and you just close the entire thing with bracket and that will be your numeric notation basically, right? So CDE is like this. So D is together and then C is together with that unit. A, B are together as a separate, kind of unit and then F is alone here. And then those are joined by the actual origin point from here. So it will be answer C, right? As said in the chat. Yeah. So we'll discuss some of those again. Uh, so we also saw some questions or some type of questions where you have to calculate the average values, right? Average hydrophobic values or something like that. Again, those are simple numeric sort of questions. For example, if you have been given these kind of values, what you just have to do is that you have to uh, put how many number of a particular amino acid, let's say, is present, then times their hydrophobic value. You have to just do a summation of that and just you have to divide by the total number, right? So, for example, in this case, if this question was asked, what you could do is that you put all of the values here, uh, and then you can just sum them all of them up, and then you divide by total number. In this case, it will give you 0.36. So you can solve those numeric questions very easily, right? Now, let's say you have a phylogenetic tree like this. Uh, so could you answer in the chat what would be the numeric notation for this one? Using the same concept by identifying the nodes that are connected through a common origin point, you can group them up and you can just proceed from that, right? So I see one answer, A and then BC together and then D together, exactly. So something like that. You can put A before BC also here. So these two are together and then A is having a common point with this unit, uh, which is, acting as together. And then again, D and E, their common point is here. So this D and E is having origin point with this ABC entire thing. So that way you can construct the phylogenetic tree or convert the phylogenetic tree to a numeric notation. Now let's say you are asked something like this. Okay, so that seems to be the same phylogenetic tree, but here some values are given. Uh, so here same evolutionary distance from a common branching point is not considered, but the distances are basically given here. Let's say in some sort of normalized way. So you have, for example, in this From this origin point, we are having B and C two nodes, right? So earlier in UPGMA, we saw that same distance was considered, but here the different distances are given. So to write when distance to convert to numeric notation when distances are given, it is the same thing. You have to just put a colon and then after that the value distance value here. So basically the same thing if you think about it, right? You have to just incorporate the distance values in the notation. Okay. Now, uh, while constructing phylogenetic trees, so there are two types of trees that we can construct basically. One would be rooted trees, another will be unrooted trees. Now, rooted trees are trees where a common ancestry point is clearly defined. For example, if we saw earlier, let's say something like this, like A, B are connected at a point together here, and then C is also connected to the A and B, and you are seeing a common, basically defined common ancestry point between this A, B unit and then the C unit also. So that will be a rooted tree. It has a root. It has a common beginning where the tree branching is starting, right? However, on the other side, you could have unrooted trees. So unrooted trees means where the common ancestry point is not defined clearly. So you do not know where the common ancestry point begins, so where the origin of all of this branching begins. So for example, if with four different taxa we constructed tree, for example, let's say A and B are connected here, and then let's say C and D are like this. However, between A, B as a unit and C and D as a unit, you do not basically know another branching point. So let's say here it was E, right? Sorry, it was, let's say, D. So let's say D was a branching point D, or you can just think of it in any way. So if you do not even put a name, that would be also fine. But basically, what is happening here is that you do not know where the common ancestry point is uh, in between this A and B and C and D as a unit, where it lies. So that will be kind of the concept of an unrooted tree. Now, you might get asked, okay, so for rooted trees and unrooted trees, how many possible arrangements are there if you are given, let's say, the species number or the number of taxa? So, how many rooted and unrooted trees will be possible if, let's say, you have a species number of 3? So, how many ways you can arrange these 3 different taxa, 3 sequences, 3 species, whatever, uh, so that they will have unrooted trees and then so that they will have rooted trees? There is a pretty handy formula for that. The number of rooted trees and unrooted trees are given by basically this formula. So here, if you have the taxa number n, basically you have to use this formula for rooted tree. So factorial of 2n minus 3 divided by 2 to the power n minus 2 and times n minus 2 factorial. So for example, okay, so using this formula, what could be the answer of this of the number of rooted and unrooted tree question for 3 taxa? So could you calculate it out very quickly and share the chat? So here, if you calculate, you will find out For example, if we calculate the number of rooted trees here, so n are, let's say we're trying to calculate, right? So since we have 3 taxa, so it will be 3 times 2 minus 3. So 6 minus 3, that will be factorial 3 divided by 2 to the power n minus 2. So n is 3, so 2 to the power 1 times n minus 2 factorial. So again, your since n is 3, so it is only factorial 1. So what it will be? So 3 times 2 times 1. Here we have 2 times 1 equals 3, right? So total 3 rooted trees you will get using this formula. Similarly, for unrooted trees, if you do the calculation, you will find that 2n minus 6 is, so 3 times 2 minus 5, so that is 1, so factorial 1 divided by 2 to the power n minus 3. Since your 3 is, n is 3 only, so it will be 2 to the power 0 times n minus 3, again 0 factorial. So 0 factorial is considered 1, 2 to the power 0 is considered 1. So it will be in overall 1, right? So the number of rooted trees will be 3, number of unrooted trees will be 1, and the 3, 1 will basically be the answer for this question. So if you are given like, let's say 4 species, 5 species, and so on, so you can just use this formula to calculate it out. Now here we have examples of how the rooted and unrooted trees are basically formed using, let's say, 3 taxa here, and then on the right side we have 4 taxa. So you can see just the arrangement, how it goes, right? So at every rooted tree you have origin like common ancestry point. For the unrooted tree, the common origin point is unknown, right? And here we have a plot where on the x-axis we have the number of species or taxa, and then on the y-axis we have the number of tree topologies that are possible. So with increasing taxa, obviously we see that the number of tree topologies will be like very high, right? And of course, the number of unrooted trees is always lower than the number of rooted trees. Okay. So this is similar to a question that we discussed earlier also. Can somebody mention in the chat quickly? Amphipathic character of amino acids are dependent on what? Based on what? Amphipathicity is basically the periodicity of polar and non-polar residues, as we discussed earlier also. So there should be a periodic nature between the presence of or the occurrence of polar, it could be uncharged polar or charged amino acids, and the non-polar amino acids, right? That would give an amphipathic nature to this stage of sequence. Now you could be asked some sort of simple numeric questions like this. For example, here you have been asked what could be the composition of alanine in this given sequence. So using let's say this formula, summation of ni times 100 divided by n, could you calculate it out very quickly? What could be the composition of alanine in this sequence? Uh, I see answer C, I guess. Something. Uh, in this particular case, since we have uh, since we have 4 alanines present out of a total number of 12 residues, so it will be just simply 4 by 12 multiplied. Sorry, yeah, 4 by 12 multiplied by 100, and that will be 33.33%. So these sort of simple numeric questions might again come in the exam or assignments and so on. So you can try to calculate it using just simple sort of formulas, right? So here we had another question. Uh, Basically, let's say you are trying to relate different uh, sort of groups of proteins, right? And you're trying to identify, okay, so you have a new protein, you already have well studied set of proteins, and you are trying to identify that which group this new protein belongs to. So in this case, this new protein will be X, right? So then, let's say you are calculating the deviation of the composition of the amino acids, and you are you're basically trying to summate every for every amino acid that is present in the protein. So from that, you are getting some sort of deviation values or some sort of sigma values, let's say, right? Uh, so the sigma values for A will be the summation of composition of I for A minus composition of I for X. And then you are calculating again sigma value for B also from protein X. And you are seeing an overall relation like this between sigma of A and sigma of B, where sigma of A is higher than sigma of B. Excuse me. Yeah. Sigma of A is higher than sigma of B. So then what you can infer from this information? Which group will protein X belong to? You have calculated the compositional deviation of A and B from X. And the sigma value or this deviation value, uh, you have some sort of relationship. For example, sigma of A is shown to be greater than sigma of B. So then if you are asked, okay, so the protein X belongs to which of these groups? What will be the answer? So in this case, of course, lower the difference or the deviation from a certain group, you can expect the new protein to be a part of that group only, right? So in this case, since the deviation from B is lower than deviation from A, you will expect that this protein should fall into group B. If we are just like using a simple example, there are only two groups, right? A and B. Uh, so it's of course, the protein will more likely belong to group B because group B has a lower sort of deviation value than group A. So this sort of reasoning type of simple reasoning type of questions might also appear in the assignments and so on. Okay, so I think for today's class we'll just wrap it up. Okay, uh, and then again we will meet in the next class, the next Friday. So please also try to attend the other TA sessions that will happen in Saturdays and Mondays, and then carefully go through Professor Pumia's lectures. And for my next TA session, it will be again in the next Friday. Okay. So thank you everyone for joining. Uh, and best of luck for the assignment questions and the exams also. Okay. Thank you.